So this afternoon, I'm going to talk to you about a project that is almost completed, so it hadn't been published, and I'm especially interested in your feedback. Okay, so before I talk about this, let's first make sure that we're on the same page on some of the terms. So I said it's strongly non-neutral. What does that mean? In population genetics, we talk about a mutation or mutant being neutral or non-neutral that is relative to the effect of drift. And the effect of drift is measured by 1 over effective population size, 1 over NE. So if the selection coefficient S is on the order of 1 over NE, we say this mutant is nearly neutral. If S is much lower than 1 over NE, then the mutant is effectively neutral. If the S is much larger than 1 over NE, then we say it's strongly non-neutral. Okay, so that is the definition. And I'm going to talk about synonymous mutations. And you all know that, okay, some nucleotide changes do not affect the encoded amino acid, like the CUU changes to CUC. It will be still using that synonymous mutation. If the CUU changes to CCU, that's protein. Using to protein, that would be called the non-synonymous mutations. So soon after the cracking of the genetic code table, people realized that a lot of mutations would be synonymous. And because they don't affect the protein sequence, the assumption is they are neutral or they are close to being neutral. So for example, in this 1968 paper by Kimura, this is actually a very important paper. In this paper, he derived the formula for the expected level of genetic polymorphism for neutral mutations on the mutation drift balance. Okay, and in the discussion, he talked about, so what kind of mutations are neutral that my formula can apply to? He said, okay, this kind of neutral. In the end, he said, still others cause no alteration of amino acids, and their mutation effect in general should be minimal. He was talking about synonymous mutations. And he went on to calculate, based on the genetic code table, what fraction of mutations would be synonymous. He said, if all mutations were transitions, then 34% would be synonymous. If transitions and transversions are equally likely, then about 23% will be synonymous. So, you know, between a quarter to a third of mutations are synonymous. This is another paper around that time by King and Jukes, where they proposed the idea of neutral evolution. And in this paper, they also said, as far as it is known, synonymous mutations are truly neutral with respect to natural selection. But we know that synonymous mutations are not strictly neutral because they could actually affect a lot of biological processes. Yet people still believe that vast majority of synonymous mutations are nearly neutral, at least nearly neutral, which is a contrast to non-synonymous mutations that alter protein sequence and often fitness. Okay, so what are the processes that synonymous mutations can affect? First of all, synonymous mutations can affect transcription factor binding, so it can affect transcription. It can also affect pre-mRNA splicing if those mutations occur near a compound rate, right? Next, it can affect mRNA folding because these synonymous mutations are in the mRNA sequence, so they potentially can affect mRNA folding. They can also affect mRNA degradation. This is a new area of research that has been recently studied by Jeff Kohler's lab. Specifically, he found that if a gene uses unpreferred codons, those codons that tend to have low corresponding tRNA concentration, then during the translation, this is the ribosome. This is the A site of the ribosome where the tRNA will come in. Okay, so if you are a rare codon, you wait for a tRNA that has low concentration, you tend to have longer waiting time. So this A site is empty for quite a long time, and the cell has a system to detect that signal. If you wait for too long, the cell will activate a complex called the CCR or NOT complex. And when this complex is activated, it will initiate the degradation of the poly A tail as well as decapping the 5'. So that will start the degradation of messenger RNA. So his group showed that when you change the codons from preferred codons to unpreferred codons, the mRNA level is reduced. And this is the mechanism they found. Okay, so this mRNA degradation. And then if the synonymous mutations happen in the first exon, for example, that's where the protein translation initiation starts. It could affect the mRNA folding in the beginning of the mRNA, often affects the translation initiation. That can also affect translational efficiency, as I said, because if you have rare codons, you tend to wait longer for the tRNA. Okay. Translational accuracy. So translational accuracy hypothesis was proposed by Hiroshi Akashi about almost 30 years ago. The idea is that rare codons, if you use rare codons, the translational error rate is high. If you use common codons, the translation is more accurate. There was some indirect evidence supporting that. And recently we found some direct evidence from analyzing E. coli proteomic data. So from proteomic data, by comparing the proteomic data with the genome sequence, you can infer the mistranslation rate. So we infer the mistranslation rate in this figure. The y-axis is the relative mistranslation rate. We are comparing synonymous codons. The x-axis is the relative synonymous codon usage. And you see there's a negative correlation. So the rare codons tend to have high mistranslation rate. And what is the mechanism of that? Well, we don't know exactly the mechanism, but we found a correlation that suggests the mechanism. So the correlation we found is here. So the x-axis is the relative ratio of the cognate tRNA concentration to near cognate tRNA concentration. Cognate tRNA is the one that matches the codon. Near cognate tRNA is the one that has one mismatch with the codon. So the idea is that cognate tRNA and near cognate tRNA would compete for this A site. Okay, so if you have a lot of near cognate tRNA, basically it will create a lot of mistranslation. So it looks like this rate, when this ratio is high, the cognate to near cognate ratio is high, the error rate would reduce. So this might be the potential mechanism for the translational accuracy hypothesis. And finally, there is the effect of synonymous mutation on co-translational protein folding. We know that protein folding actually occurs during translation. So if you have a domain of protein that has been translated, this domain would fold while the second domain is still being translated. And people have found that rare codons tend to appear between domains. And the idea is that the rare codon between domains would basically pause the translation, and this gives time for the folding of the already translated part of the protein. So if there is a mutation there, that could affect co-translation of protein folding. Okay, so there are a lot of ways that synonymous mutations can affect biological processes and potential fitness. And empirically, we actually have some evidence that synonymous mutations can be strongly non-neutral. Here's one example. In this paper, we're talking about two mutations here, M3 and M4. Those are synonymous mutation, and then there is another mutation called M1, which is in the promoter. So if you look at this figure, which is the growth rate of the bacteria, the M1 is here. Okay, so the growth rate is like 0.3. And if you look at here, M1, M3, that is on the background of M1, you add mutation M3. You can see that the growth rate increased a lot, more than 10%. right? And this is M1 plus M4, which also increases much relative to M1. So there's anecdotal evidence that synonymous mutations can have quite drastic fitness effect. This is an early paper by Lind et al. They systematically mutated 2 ribosomal protein genes in Salmonella. They made 38 synonymous mutations and 88 non-synonymous mutations individually. And then they measured the growth rate of those mutants relative to the wild type. And you can see here the 2 distributions, the synonymous and non-synonymous. The 2 distributions are very similar, and actually not significantly different. So the top panels are in LB, the lower panels are in M9, a different medium. So in both cases, there's no significant difference in distribution of fitness effect between synonymous and non-synonymous mutations. But they believe that this is specific to ribosomal protein genes. And the reason they think so is because ribosomal protein genes are extremely highly expressed. They are among the most highly expressed genes in the genome. And also they are folding, mRNA folding is special. So they actually think the reason that synonymous mutations affect fitness in this case is because of the effect on mRNA folding. More recently, Zhang et al. Had this study where they compared the 2 yeast strains, EY, which is a lab strain, and IM11, which is a vineyard strain. And they identified all the single nucleotide polymorphisms between them, thousands of them, and then used a CRISPR-Cas9 based method to engineer those mutations on the background of DY to see whether they have fitness effects. So the blue bars show the mutations they actually tested, okay? And the brown bars show the mutations that they found to have significant fitness effects. So if you look at the synonymous, the top is synonymous, second is missense, that's non-synonymous. You see that the ratio between the synonymous and the missense for the brown and for blue are very similar. That means the same fraction, similar fraction of synonymous SNPs and non-synonymous SNPs are having fitness effect, okay? The caveat here is that polymorphisms may not represent all random mutations. They have been through selection, right? So related adherence mutations have been removed already. But at least it shows a lot of synonymous polymorphisms are having fitness effect. And on the same year, there was another paper that tried to do QTL mapping of growth rate traits in yeast between IM11 and YJM975. YJM is a clinical strain. And they designed this QTL mapping that is high resolution. They could map the causal mutation to a single nucleotide. Okay, so they were able to tell whether that whether that SNP is a missense or synonymous, etc. So they found a lot of causal mutations that are synonymous, so shown here by the black bar here. And also, when they compare the effect size, they found that non-synonymous mutations and the synonymous mutations, the effect size are not very different. Okay, but again, this is a polymorphism. They are not representing all mutations, but still indicating a lot of synonymous polymorphisms have fitness effects. Because pH trait is growth rate, right? That's fitness. So the available empirical evidence suggests that at least some synonymous mutations are strongly non-neutral, but how widespread this phenomenon is remains unclear. So our objective is to test the neutral or nearly neutral hypothesis of many synonymous mutations in a diverse set of genes. Okay, so we're going to use the budding yeast Saccharomyces cerevisiae. We previously developed a method for high throughput measurement of fitness of a large number of mutants in yeast. And I will explain that method in a minute. So what genes should we choose? We have several criteria. First, we want our genes to cover a lot of different biological processes. Second, given that there is a strong correlation between gene expression level and the synonymous codon usage, we want our genes to cover a whole range of expression level so that we can see whether the fitness effect has anything to do with expression level of the gene. Okay. We want the genes to be non-essential because one of the steps is to delete the gene. So we can't delete essential genes. So it has to be non-essential, but it has to have fitness effect when we delete the gene, because if the gene has no fitness effect when you delete it, it's unlikely that a point mutation would have fitness effect. So these are all criteria. And in the end, we picked the 21 genes. They cover a lot of biological processes such as metabolism, coating, remodeling, transcription, translation, cell wall synthesis, etc. Okay. So this panel shows the distribution of the expression level of the 21 genes. On the top of the panel, these are the log2 mRNA level in RPKM. So the lowest expression is 3, the highest is 13. So that is about 1,000-fold expression level difference. The bottom of this graph shows the ranking of the endogenous genes in yeast. This is the most highly expressed gene. This is the most lowly expressed gene. So our genes are mostly actually in the top half. There are a couple of genes that are in the bottom half in terms of expression level relative to all the genes. And the reason of this bias is very lowly expressed genes cannot have fitness effect when you delete them. So we had to choose the genes that are relatively highly expressed. But still, the genes we choose cover about 1,000-fold of expression level. So this is our experimental procedure. For each of these genes, we randomly pick a 150 base pair coding region. And for each position, we can mutate it from the wild type to the other 3 nucleotides. So we can create 450 single mutation variants, right? So we would replace the endogenous, actually the 150 nucleotide segment in the endogenous gene with the synthesized variants, okay? So here, and then we would add in the mixture the wild type. So this is a library of theoretically 451 genotypes. And we compute them all in the same test tube for 48 hours, okay? And we got this new population. And from each population, we would amplify this segment and then sequence it by Illumina. So the Illumina sequencing provides 2 pieces of information. One is the sequence, so the genotype. Second is the genotype frequency. So by comparing the genotype frequency between T0 and T48, we can calculate the relative fitness of the genotype relative to the wild type. For some of the mutants, their fitness is really low. So by 48 hours, they are gone in the population. So we added a 12-hour time point that is specific for calculating the fitness for those related curious ones. Okay. So that is our procedure. And in the end, we got the fitness estimates for about 8,000 mutants. We apply them to have at least 50 reads at T0. That is to ensure a certain level of accuracy of fitness estimate. So this includes about 1,800 synonymous mutations, 6,000 non-synonymous, and 169 nonsense mutants. Okay, so the first step we did was because we didn't observe all of the mutants. Okay, we asked the company to synthesize them, but we didn't observe all of them. So it could be that the company didn't synthesize some of them. It could be they are really deleterious, so they didn't appear in our library. So we want to see whether there's a bias. And you can see here is the observed mutants on the right, and on the left is the unobserved mutants, according to our design. So there is no significant difference in terms of synonymous non-synonymous ratio between them. So that means it's not biased, at least for that part. The fitness measurement is relatively accurate. You can see the replicates, the correlation is good. So we also independently synthesize a subset of mutants and measured their fitness by monoculture growth rate. So the x-axis here is the growth rate based fitness, and the y-axis is the sequencing based fitness, and there are about 10 of them. They are all synonymous mutants. Okay, so the two methods give good correlation in terms of their fitness. So our fitness is measured in YPD at 30 degrees. YPD is a commonly used rich media for yeast in the lab. And whether that fitness measured in the lab has any biological or evolutionary relevance. So we compared with evolutionary data. We had an alignment of 6 species that are all after the whole genome duplication in yeast evolution. And we looked at the orthologous protein alignment and the amino acid positions that are variant versus invariant. The prediction is that if the amino acid position is invariant, the non-synonymous mutations at those positions are more likely to be deleterious. So that is indeed the case. We found a significant difference in fitness. So that tells us at least the fitness measured in lab is somewhat, at least somewhat valuable. Okay, so now let's look at the fitness distribution of those mutants. This is the nonsense mutant. Okay, and this distribution is weird, but we have only 169 mutants, so it's probably idiosyncratic. And these are the synonymous and non-synonymous. So the blue is non-synonymous, yellow is synonymous. I'm going to use this color scheme throughout. And when you look at the distribution, they are extremely similar. So the median, one is 0.988, the other is 0.989. The mean, 985, 988. So they are very similar. So the majority of the mutations mutants have fitness slightly below 1 for both non-synonymous, synonymous. There is a tail you can barely see here, and you can see here, but the tail part looks like the blue has a bigger tail. Okay, so when you test the difference between the two, they are significantly different. Although by naked eye they look very similar, they are significantly different. And the difference actually is around here from 0.91 to 0.99. That range has difference. So it looks like from 0.91 fitness to 0.97, there are more non-synonymous mutations. From 0.97 to 0.99, there are more synonymous. Okay. Now, when we look at the 21 genes individually, most of them do not show significant difference between synonymous and non-synonymous fitness effects, but a few of them show significance. These are indicated by the red stars. But even in those cases where there is significant difference, when you look at them, the blue and the yellow, you will see blue and yellow are pretty similar. Yellow is actually quite distinct from the fitness of 1, which is this dotted line. So you look at those significant cases like here, for example, the yellow is far from 1, but it's quite similar to the blue. Okay, in those cases that they're significant different. Okay, so basically all 21 genes show the similar pattern, that is synonymous fitness effect and non-synonymous, very similar. So if we have 4 replicates in the competition. So our fitness estimate has standard error. We can test whether a mutant has fitness significantly different from 1. We divide it into 3 beings, significantly lower than 1, significantly higher than 1, and neither. And you look at the distribution for synonymous and non-synonymous, there's no significant difference. About 75% of synonymous mutations and about 75% of non-synonymous mutations are significant deleterious. About 1% are significant beneficial. So we found that in among all of our mutants, the smallest fitness effect that is statistically significant is 0.001. 0.001, that S is much, much higher than what natural selection can detect. In yeast, that is 10 to minus 7. So all of those significant fitness effects are strongly non-neutral. Now we want to understand what is causing synonymous mutations fitness effect. The first thing comes to mind is codon usage, okay? And we know highly expressed genes have more biased codon usage. So presumably, synonymous mutations in highly expressed gene would be more deleterious. And so the x-axis in this graph is the expression level of the gene. The y-axis is the mean fitness of the synonymous mutants in that gene. And we're expecting a negative correlation. We did find a negative correlation, but it is not significant. And we think actually also, if we remove this dot, probably it's not even negative. So basically this negative correlation is very weak. It may not even exist. The other thing we consider is non-synonymous mutations and synonymous mutation have such similar fitness distribution. That seems to suggest to us that perhaps the reason that non-synonymous mutations are deleterious is not because they affect protein sequence, because otherwise they wouldn't be so similar to synonymous mutation. Instead, maybe synonymous mutation, non-synonymous mutation, their fitness effects are all opposed due to another factor that is shared between synonymous and non-synonymous mutation. And that thinking leads us to considering gene expression level. Synonymous mutations and non-synonymous mutations, they are both in the coding region, and both of them can affect gene expression level. So we next wanted to measure the expression level of this mutated gene relative to the wild type version. Okay, so I'm calling this REL, which is the relative expression level of a mutant to the wild type. Okay, so here's how we measure it. So remember, we have this pool of the wild type and 450 mutants of a gene. We do a DNA seq, okay, which tells us of the frequency or relative concentration of these genotypes. We also do a RNA seq, okay, we extract the RNA and do an RNA seq, which tells us the amount of RNA of each genotype. So we divide the RNA level by the DNA level. That tells us the expression level of this gene in each mutant. So we did that, and we did a lot of replicates. This is showing the correlation among replicates, which is about 8.85. Okay, so this is our result. So this is a distribution of the relative mRNA level of non-synonymous mutants. So the lowest is almost 0. The highest is more than double the wild type expression level, and about 55% of the non-synonymous mutants significantly affect the expression level of that gene. For synonymous, it's very similar. The distributions are not even significantly different. This is interesting. We also have nonsense mutations. So for nonsense mutants, their expression level is very low, and that is because of nonsense mediated decay. In eukaryotes, that's a mechanism to degrade the mRNA when the mRNA contains premature stop codon. So you can see all of them have really low mRNA level. Okay, now we want to see whether the effect of those mutations on the mRNA level is the cause of their fitness effect. Okay, so that requires us to compare those mutants across different genes. And when you delete a gene, the fitness effect depends on the gene. Some genes, when you delete, have little fitness effect. Some of them have big fitness effect. So to allow comparison among mutants across genes, we calculate something we called rescaled mutant fitness. So this is a fitness of 1, this is 0, and F0 is the fitness of the gene deletion string for that gene. So gene deletion is equivalent to reducing the expression level by 100%, right? So if we want to see what is the effect of changing the expression level on the fitness, basically we're looking at in this way, from 1 to fitness of 1 to, it could be high, okay, to the lowest is F0, which is when you delete the gene. So we calculate this rescaled fitness, okay, by this formula. Basically, it's this relative to this way, okay, and then we compare across all the mutants among all the genes. So this is what we found. On the left, you can see these are non-synonymous mutants. The x-axis is the relative expression level. The y-axis is the rescaled fitness. So there is this positive correlation, but there's a difference when the expression level is below the wild type versus higher than the wild type. When it's below the wild type, there is a positive correlation. When you reduce the expression level, fitness is reduced. Above the wild type, there is very little correlation. So when you increase expression level above the wild type, seems there's very little fitness effect. It's flat. And the same pattern is shown for synonymous mutations. So what is the mechanism that coding mutations affect the mRNA level? One possibility is they affect the binding, TF binding. So we looked at the TF binding sites within and outside the TF binding sites. So you can see here the comparison. The left bar is when the mutation occurs within a TF binding site of this gene, and the right is outside. And the y-axis is the absolute value of the fractional mRNA level change caused by the mutation. We don't see difference. We don't see significant difference for both non-synonymous and for synonymous. So that indicates that the reason that coding mutations affect mRNA level is probably not through affecting TF binding. Okay. And if you remember, I mentioned that Jeff Coller's work showing that there is a mechanism in the cell that can affect the mRNA concentration because of degradation. And he showed in a manipulative experiment that when he changed rare codon to common codon and changed from common codon to rare codon, the mRNA level is changed. Particularly, he used this codon adaptation index as a measurement for whether this is rare or common. So we also took that measurement. So here the x-axis is the relative CAI of a mutant relative to the wild type, okay? And the y-axis is the relative expression level of the gene in the mutant. So we do see a positive correlation for both non-synonymous synonymous mutants. So that indicates that the mechanism that was discovered in Jeff Coller's work probably is responsible for what we are seeing. That is the regulation of mRNA level through this CCR4 NOT protein complex that is monitoring the protein translation, whether you're waiting for the tRNA or not. So this is probably the mechanism behind our observation. The other thing we observed is that for both synonymous and non-synonymous mutations, there is a correlation between the expression level of the gene and the fraction of mutations lowering CAI. That is expected because highly expressed genes have high CAI. So mutations were more likely to decrease CAI. And lowly expressed genes have low CAI, so mutations would tend to increase CAI. So there is this correlation, and this correlation is translated into the correlation between expression level of the gene, the fraction of mutations lowering the expression of the gene, which is then translated into fitness. So in a more highly expressed gene, a random synonymous mutation is more likely to lower the fitness. And that is also the case for non-synonymous mutation. So in a more highly expressed gene, by the way, I should say this is the rescaled fitness. For in a more highly expressed in non-synonymous mutation, also more likely to reduce the rescaled fitness. Earlier I mentioned the Lind et al. Paper in which they showed that in ribosomal protein genes, synonymous mutations and non-synonymous mutations affect fitness probably through affecting the MI folding. So we checked the MI folding. This is predicted, computationally predicted MI folding. There's a little bit of correlation. The XX here is the relative folding strength of the mutant to the wild type. And y-axis is the rescaled fitness. So there is significant but quite weak correlation for both non-synonymous and synonymous. So the correlation is much weaker than what we see in terms of affecting expression level. So that is to say the fitness effect is more likely caused by the effect on mRNA level rather than on mRNA folding. Okay, so we're facing this difficult question. The non-synonymous and synonymous mutants show similar distribution of fitness effect. Yet when you compare different species, you calculate the non-synonymous substitution rate and synonymous substitution rate. This DN to DS ratio is usually much lower than 1 for the vast majority of genes in the genome. If they have similar fitness effects, why is the ratio significantly lower than 1? We have two possible explanations. The first explanation is that our measurement of fitness effect, the distribution of fitness effect, has a limited resolution. Okay, so basically, fitness effect smaller than 0.005 is often not significant in our experiment. That means our resolution is around 0.005. So although the distribution looks very similar at this level, we see, but we actually don't know the distribution in this range, which, for example, in this range, from 0.995 to 1, this fitness range, our experimental method has no power to measure. Okay, so we actually don't know in this range how the distribution look like, whether they are same between the two. So for yeast, effective population size is 10 to the 7th. So anything fitness lower than 0.99999 would be deleterious and selected against. Okay, so if in this range, which is kind of a neutral range, in this range, the fraction of non-synonymous mutation is 10% of the fraction of synonymous mutation, then we will observe at the NDS ratio of 0.1. But whether that's the case in this range, our method cannot tell. Okay, so I think this remains a viable explanation of still why the NDS ratio is lower than 1, is much lower than 1, despite that our two distributions measured in the lab look similar. Okay, so that's one potential explanation. An alternative explanation is that we measure the fitness under only one condition. Okay, and there have been some reports that amino acid changes often have different fitness effects in different conditions. Now, if synonymous mutations do not have variable effects on the different conditions, but non-synonymous mutations have variable effects on the different conditions, then that difference could cause a DNDS ratio that is smaller than 1. And we have to consider multiple environments because the fixation time for a mutation is actually very long. For neutral mutation, takes about 4 N generations. Okay, so that's a very long time, and the environment is unlikely to be constant during that time. So in a more extreme case, if you look at this diagram, so we have 2 non-synonymous mutations and 2 synonymous mutations. So 50% of them are deleterious, which is marked by this bold circle, and 50% of them are neutral. And same for synonymous mutation. So that's in environment 1. I have 4 environments. So if the fitness effect of a mutation for non-synonymous mutation varies across environments, so the same mutation in some environments is neutral, some environments deleterious, okay? But for synonymous mutation, if it's relatively constant, the same mutation is deleterious across environments, same mutation is deleterious is neutral across environments. Then the outcome here is that these 2 non-synonymous mutations both will be purged by purifying selection. But this synonymous mutation actually can be fixed. Okay, so although in each environment, you will see 50% non-synonymous mutation deleterious and 50% synonymous mutation deleterious. But when you look at 4 environments together, actually the DNDS ratio could be smaller than 1. Okay, so that is our thinking, and we want to test whether this is a viable model. So what we did is first we did a computer simulation. In the simulation, we used the fitness effects we estimated in YPD, assuming that is the true fitness effects. But then we consider multiple environments and allow variation of fitness effects among environments. And we give high variation for non-synonymous and synonymous mutations. And you can see in this plot, the y-axis is expected DNDS ratio. So if you have only 1 environment, the expected DNDS ratio is around 1 because the 2 distribution very similar, right? But as the number of environments increases, the DNDS ratio decreases. Okay. That is because we assumed non-synonymous mutation have high CV in fitness across environments. So we want to test is that true? Okay, so we measure the DFE distribution fitness effect in 3 additional environments. These 3 are more stressful environments compared to YPD. This is 37 degree. This is a H2O2 oxidative stress, and this is ethanol instead of glucose and carbon salts. And in each environment, actually, the non-synonymous synonymous distributions are remarkably similar still. Okay. And this is showing the cumulative distribution. They are significantly different. In each case, they are significantly different. And the difference you can see is around here. And if you calculate fraction of mutations that are significantly non-neutral, here it's like 50-some% significantly non-neutral. Each environment is like 50-some to 60%. And this fraction seems lower than in YPD. The only reason is that we had 3 replicates here instead of 4 replicates in YPD. So the power is reduced. So when we downsample our YPD data to 3 replicates, we observe very similar results. So this is the CV we calculated for non-synonymous mutants. Their CV is significantly higher than the synonymous mutants. And this is the y-axis is a log scale. So actually the mean CV for non-synonymous is 0.0163, for synonymous 0.0124. So there is a quite big difference in the CV. Yeah, I'll skip this. Now, if we plug in measure the fitness effects in each of these 4 environments, and then based on that, if we predict the DNDS ratio for individual, if we consider individual environments, this would be the DNDS ratio. And here we consider different fitness cutoffs. If the fitness of a mutant is lower than the cutoff in any environment, then this mutant is purged. Okay. Now, if we consider a population that rotates among the 4 environments, and if in any environment the mutation has a fitness lower than our cutoff, then we will purge that mutation. Then the DNDS ratio is lower. Okay. We also did a more rigorous way of this analysis. Now, if you consider a population going through multiple different environments, the overall fitness is a geometric mean of the individual fitness. So if you consider that and you calculate fixation probability using Kimmel's formula, you can also calculate the NDS ratio. So, and here we looked at different population size. So basically, population size doesn't matter so much, looks like. So if the population goes through multiple different environments, the DNDS ratio is indeed lower than when it's in one environment. Okay, so this is another possible explanation. And it's possible there's two explanations are both correct. They are not mutually exclusive. Okay, so in summary, I showed you that characterization of the distribution fitness effects of thousands of coding mutations in diverse yeast genes on the 4 environments show that on any environment, most synonymous and most non-synonymous mutations are strongly non-neutral, and the DFEs are overall quite similar. And our results also suggest a general mechanism through which coding mutations affect fitness, which is disturbing the mRNA level of the mutated gene. Previous studies showed that the mRNA level of a gene has a strong influence on the evolutionary rate of the protein sequence. And several mechanisms have been demonstrated. And our results actually suggest a new mechanism. So I'll just explain the new mechanism here. So here showing gene expression level. Some genes have low expression level, some genes have high expression level. And in a highly expressed gene, the fraction of mutations decreasing the mRNA level would be high. And because they are deleterious, so the fraction of mutations that are deleterious would be high. So these mutations are purged. So the fraction of mutations that are accepted, in other words, the evolution rate is lower. So this is an additional explanation for the so-called ER anticorrelation evolution. So ER means expression level, E is expression level, R is evolutionary rate. So people observe this negative correlation across a whole range of organisms. And our result provides an additional explanation of why this can happen. So we know that a lot of biological conclusions are based on the assumption that synonymous mutations are neutral. So if they're not, then we need to re-evaluate those conclusions. First of all, a lot of selections tests are based on comparison of synonymous and non-synonymous mutations. So one idea is that, okay, even if synonymous mutations are not neutral, but if we consider only the selection on the protein part, protein sequence part, and maybe for everything else, synonymous and non-synonymous, their effects are canceled out. So if non-synonymous substitution rate is still different from synonymous substitution rate, then that difference must be due to the protein level selection. That is a good thinking, but we actually found in a previous paper that there are some strange things that make synonymous and non-synonymous mutations on non-protein level effect not canceled out. For example, their effects on mRNA folding actually out. And that has to do with the GC content at the third codon position being different from the first 2 positions. Okay. And the synonymous mutation mostly at third codon position. So their effects on mRNA folding, for example, is actually not canceled out. So I think it's more complex, and we need to figure out ways to test the selection in the context that synonymous mutations are not neutral. And a lot of studies about mutation rate, mutation pattern, and mutation mechanism are based on synonymous mutations. So that needs to be reinvestigated. Effective population size is often estimated from synonymous polymorphism. So that also needs to be reinvestigated. Some molecular dating is based on synonymous mutation. Say you date the time of duplication of a gene, and you compare their synonymous distance. So that might not be correct. And finally, in studying the disease mechanism, people actually focus on non-synonymous mutation. For example, the exome sequencing. After exome sequencing, they usually only look at the non-synonymous mutation. They ignore synonymous mutation completely. But if synonymous mutations and non-synonymous mutations have very similar fitness effects, that also implies synonymous mutation can also cause disease, right? So, so that needs to be studied. And currently, there are a lot of programs that predict the potential disease consequence of non-synonymous mutations. And we need to do the same for synonymous mutations. So actually, people have looked at that. And so in about 2 years ago, this review paper already listed more than 30 diseases known to have been caused by synonymous mutations. And probably there are many more. There are some outstanding questions. How general is our finding? We did this in 21 genes, and they tend to be biased toward highly expressed, okay, in yeast. So whether it is general, how about really lowly expressed genes, whether there's smaller effect. And this is in yeast, which has very big population size. How about in vertebrates and in mammals? We don't know. And the second is that we did this work in haploid yeast. Okay, so there is no dominance recessiveness involved. And presumably most of the mutations are recessive. And whether synonymous and non-synonymous mutation will have different levels of dominance, we don't know. We are currently studying this problem. Number 3 is the mechanism of their fitness effects. We found the effects through affecting mRNA level, but is that the main effect? We don't know. That's just one of the effects, perhaps. Okay. So we still have some outstanding questions that are worth studying. And in the end, I want to thank the people who did the work. The vast majority of this project was done by graduate students, Zhang Shen. And Siliang Song is another graduate student who helped with the simulation in the last part of the talk. And Chuan Li was a past graduate student, and she helped of this experiment. Thank you all.
Okay, so maybe we'll take questions in person first, and then if you're watching us online, maybe you could put your questions in the chat, and we'll get to those next. Questions for John?
How exactly what? So it's through competition. So we have a group of genotypes in the same pool, right? And they will compete with each other in growth. And then at 2 time points, we basically, by sequencing, we measure the relative frequencies of the genotypes frequencies. Okay, relative frequency of those genotypes. So at 2 time points, you will see the frequency change relative to the wild type. And from that, you can estimate the fitness. Yeah. Yeah. It's about 90 minutes per generation.
So, my understanding that substitution rate for fluctuating environment models like the SAP model is very sensitive to the rate of fluctuation. In the simulations, did you look at different cycling patterns?
No, we haven't. We're basically assuming each environment has the same duration. So then the overall fitness is just geometric mean of that. And I guess we are assuming that each environment lasts for short time. Yeah. Yeah. Thank you.
I'm wondering if you parse out single amino acids.
We haven't done that analysis. But previous studies, I think, showed in some early studies where they did a large scale experiments where they do mutagenesis and measure the fitness effects. Most of those studies were done in virus, and they did observe that. So if the amino acids are similar in physical chemical property, usually fitness effect is smaller. But we haven't done that in our data. Yeah. Right. What we did was on non-essential gene. We cannot do essential gene, right? So I cannot comment on essential gene, but on the other hand, it seems if the mechanism, which is like through affecting expression level, if the mechanism is general, I suppose for essential gene, you would also observe that. What's the other part of your question? That I don't know. I think I mean previous studies were focused on non-synonymous mutation. So they were mostly done in virus, and they mutate the usually at the non-synonymous sites, and they look at the fitness effect. I don't know whether there's correlation with the genome side.
Variety of ways. So you're talking which is the role from the professor.
Which type of synonymous mutation?
Yeah, so on the role.
Right. So from the correlation, it looks like the CAI is correlated, right? If you change the CAI more, if synonymous mutation change the codon adaptation more, then its effect on mRNA level is bigger. But also remember that we found if the mRNA level is increased above the wild type, then it doesn't seem to have any fitness consequence. But if it's lower than wild type, then it's bad. Yeah. The size of the RNA, you mean the size of the gene, the size of the protein? We just required the protein to be at least 150 nucleotides long. Other than that, we didn't correlate. We didn't didn't check that. Yeah. Yeah. This is a randomly chosen 150 nucleotide region. Yeah. So when you say previous studies found the fitness effect to be small, they are based on what? Mutagenesis. Because a lot of studies were not measuring fitness, they were measuring biochemical activity. So the large scale measurement of fitness of point mutations are not many, and especially for synonymous. Yeah. Yeah. Haven't tried that, but do we know the binding affinity? Do we have the information about that? Right. Yeah. I understand your question. I just don't know whether there is information about the affinity. If there is, we can certainly incorporate that. Excuse me, can you say it again? Which correlation you're talking about? Around here? Yeah, this figure is actually very difficult to see because the dots are on top of each other. So it's difficult to see whether this, how many dots are there. But this red is the equal size bean. We have 10 beans. Each bean has equal number of dots. So if you look at this red dots, and they show this kind of a regression. Yeah. Oh, okay. Right. So we also did this analysis for individual genes. And for individual genes, of course, the statistical power is reduced, but we do generally see this kind of a trend. You mean some genes tend to be sensitive, some genes are not sensitive. Yeah, I don't recall seeing that, but it could be we didn't pay attention. Yeah, I don't know. Okay. So the last question, do the genes that show a significant difference in fitness effects between S and NS have more curated protein-protein interactions? Is there something else common to these genes and absent in the genes with them? Yeah, I think there are only 5 genes where the synonymous and non-synonymous distribution are significantly different. And so the sample size is very small, so it's difficult to tell what's the cause. If there's anything, it's really because those 5 genes tend to have more mutants measured for fitness. So sample size is big. I think that's the first explanation. We haven't been able to find other significant correlations. So the other, there's another question.
The other one is from Nicole Perna, who mind is blown. Any thoughts on alternate ways to develop the question that we've been
That's non-coding. But of course, non-coding is also not completely neutral, right? They could have regulatory sequence, etc. But presumably, in human and in big genomes, there are, I suppose, there are lots of non-coding regions that are almost neutral. But the question is to find which part is neutral.
Lucky enough to have
Yeah. Some introns, yeah. The introns could have regulatory sequences too, yeah.
Thanks for a great discussion. Thanks for.
Thank you.